Okay, thank you, Patrick. Thank you, Mr. Baumann, for your very kind words. Receiving this prestigious award is a great honor for me. Thinking of the people who received it in the past, the great scientists, as well as thinking of other people who actually also would be deserving this award, and then receiving it yourself is just very touching, and I'm very, very grateful for that. I'm of course also grateful to the Bayer Foundation for selecting me, as I already said, and to my teachers, Helmut Schwarz is here, as well as, most importantly, my co-workers, whose ingenuity, hard work, and dedication has made all of this possible. We cannot have yet a celebration that would be appropriate for this kind of award back in Heidelberg, but we will do this as soon as the Corona crisis allows us to do so. I have now the opportunity to tell you in my own words what I'm actually doing. And as Patrick already said it much more eloquently than I will be able to do this, is what we're trying to do is to visualize biology with chemistry. And in my presentation, what I'll be trying to do is to give you a historical perspective of the work that we're doing, and then explain to you through a number of examples what it is, what we do. Chemistry has always been a driver for pushing biology forwards. And if we go back 150 years, we come to the work of Paul Ehrlich that Patrick also already mentioned. And Paul Ehrlich, early in his career, was obsessed with using dyes to stain tissues and cells and to discover new biology. And there's a famous saying of the lab mates of Paul Ehrlich who witnessed his obsession that can unfortunately not translated into English. Ehrlich färbt am längsten. And as one of the examples that the work of Paul Ehrlich, I would like to introduce to you this dye monophenylrosanilin that Paul Ehrlich at the time received from the German chemical industry that developed at the time. And then he used this monophenylrosanilin, or in short dahlia, to develop a staining procedure that allowed him to identify mast cells, a new cell type that he actually called mast cells. And I find this is a fantastic example, this manuscript. You can see here a figure from the original manuscript where he depicts the mast cell. It's a fantastic example how chemistry can be used to develop or to discover new biology. However, Paul Ehrlich probably would not have been able even to imagine now the kind of resolution that we can achieve when we now today use chemical dyes and various methods to stain proteins in live cells. And as an example, I would like to show you two movies, micrographs, of cells that we labeled with different chemical probes that selectively attach themselves to various proteins in the cell. And if you just look here to the image of the movie to the right, what you'll be seeing is these red little dots. These are lysosomes that are moving along the tracks of microtubuli. And these kind of experiments reveal a lot about the inner work of a cell. And so you might ask yourself, how can you achieve such a specific labeling? How can you make sure that the dye that you're interested in attaches itself only to that particular protein? About 20 years ago, when we were facing this problem, we introduced what is now called so-called self-labeling protein tags. So let me explain to you what that is. A self-labeling protein tag is a small peptide or protein that you genetically fuse to the protein of interest. And what the role of this tag is to undergo a specific, highly selective reaction with the chemical probe that you add to the cells, such that the chemical probe covalently and irreversibly attaches itself to the tag and thereby labels the protein of interest. The specificity of the approach thus comes from the genetic engineering of your protein of interest, as well as this highly specific chemical reaction between the tag and the chemical probe. And the challenging part at the time actually was to identify chemical reactions that are selective and fast enough to achieve this kind of selective labeling in live cells in the time span of minutes or maybe also hours. So what we were looking for was a tag that undergoes such a highly unusual reaction. And the protein that caught our attention was a DNA repair protein. the O6 alkyl guanine DNA alkyl transferase, in short AGT, that Patrick also already mentioned. And what AGT does is it repairs a DNA lesion, the alkylation of guanine. And you can see here in this cartoon a methylated guanine, that methyl being an alkyl group, that then is removed in a highly specific and fast reaction by this AGT that transfers the alkyl group to a reactive cysteine residue. What is unusual in this reaction is that the AGT is actually not an enzyme. It just does one reaction. It alkylates itself, and that's it. It sacrifices itself for the good of the cell. And so if you think about this, nature evolved here a protein whose goal is to alkylate itself. If we think about this in the context of protein labeling, that sounds very much like what we are looking for. So what we did is, with a lot of engineering of the protein itself and the design of appropriate substrates, we designed the so-called Snaptech, which is an engineered alkyl guanine transferase that now, instead of repairing DNA, reacts with these benzyl guanine derivatives that carry a chemical probe, thereby leading to a covalent and irreversible labeling of the Snaptech, and thereby your protein of interest. Snaptech labeling is as easy as 1, 2, 3. What you have to do first is you need to genetically engineer your cell, transfect the cells that they express this fusion protein. Then you just pipette the benzyl guanine molecule into the solution, which then diffuses into the cell and is snapped up by Snaptech. And therefore then leads to the labeling, the fluorescent labeling of your fusion protein, which you then can analyze or image through fluorescent microscopy. So it's a very easy procedure, but what this cartoon actually doesn't depict is that when we introduced the Snaptech and solved this first problem, we actually run into another problem. And that is that most of the fluorophores or probes that we would like to use for the labeling actually do not like to go into cells. And to show you this is I depict here a number of dyes, fluorescent dyes, that biophysicists love to use. These are dyes that have high extinction coefficient, high quantum yield, are very photostable, and emit and are excited in the far red spectrum of light. However, if you look at these molecules, their spectroscopic properties are due to these extended aromatic systems. These are very hydrophobic molecules, and then to keep the molecule in solution, what you add is sulfonic acids to keep them charged and not crashing out. And these molecules are exquisitely membrane impermeable. So for us, that was always an enormous source of frustration. We developed this cool tool, but then the dyes that we really wanted to use, they just didn't get into the cells. And so that was really a sense of frustration. And it also was a call for more chemistry. So what we need is dyes that actually also are biocompatible, as we call it. We then, that was about 10 years ago, stumbled about a class of dyes, the silicon rhodamines, that provided a very interesting solution to that problem. Silicon rhodamines are shown here on this slide. And what this molecule does is it exists in a dynamic equilibrium between two forms. What you see on the left is the Zwitter ion. It's polar, and it is the fluorescent form of the molecule. This molecule, however, also can form a so-called spirolactone, which is shown in black on the right. And this molecule is non-polar, non-charged, and non-fluorescent. You might now say, why would I want to have a dye that is partially present in the non-fluorescent form that can only reduce my brightness? However, the advantage of using these kind of molecules is that the spirolactone is a molecule that is not charged, not polar, passively can diffuse into the cell. And then that was really where we got lucky here is the undergoing of the labeling reaction pushes the equilibrium back to the open state that introduces the fluorescence. That is, there is fluorogenicity, as we call it. The molecule becomes highly fluorescent only upon binding to its target. So you have a molecule that then when you add it in combination with various labeling methods, before it binds to its targets, doesn't really contribute to the background. When it binds to its cells, it goes to the fluorescent state and then allows you for the imaging. And I would just like to show you a few micrographs of experiments where we used a combination of different probes based on these principles with different colors that we just add to cultures of cells. They diffuse into cells. You don't even have to wash them away, and they highlight the structure that they're supposed to bind to. This kind of technology has become very popular in the life sciences because of the ease of how you apply it, as well as the quality of the images, the information that they reveal. And since the introduction of both Snaptech and these various dyes that we have made, there are hundreds of publications of other scientists that use these technologies. One of my favorite publications is this one here that describes the use of one of our probes on the International Space Station, which for a short time increased the standing, my standing versus my two sons at home, because they thought that actually what I do maybe is interesting. But jokes aside, the distribution or the impact of the tools that we have developed very much depends on the commercial availability of these molecules, because scientists, being them on the International Space Station or whatever, what they need to do is they have to be able to purchase these molecules. And I have been very fortunate, that has also already been mentioned, to be involved in the founding of 2 startup companies that commercialize the labeling technologies that my laboratory has developed. I'm extremely grateful to my partners in crime in these endeavors, and that has greatly increased the impact of our work. I would also like to show you another example of how you can use this protein labeling for not only visualizing where a protein is, but also learn something about biology. For example, how a biological structure is built. And the example that I would like to discuss with you is tracking cell wall growth in budding yeast. What you see here on the left is a cartoon of a budding yeast cell. That is, budding is the process where a daughter cell is originating and then finally will separate itself from the mother cell. What differentiates us from yeast cells, among other things, is that yeast cells have the cell wall that protects them. And that cell wall is a structure of carbohydrates and proteins covalently attached to the cell wall. And so let's take one of these cell wall proteins and provide it or label it with a tag, and then use a fluorophore to label the cell wall green. As you look at the cell, the budding process is not yet finished. So what the yeast cell then will need to do in order to finish the budding or proceed with the budding is the bud needs to grow, and it needs to make new cell wall structure that was not present in the first part of the experiment. So then what we can do is we come with another different color dye, add it to the yeast cell now, and now this new part of the cell wall is stained red if you're using a red dye. Again, you can continue this by using a blue dye, for example, to visualize then how the daughter and the mother cell are separated, sealed off by the synthesis of cell wall and membrane structure between the two. And if you now look at the micrograph of the cartoon that I show you, what you really can see is that by using the sequential labeling with different chemicals that selectively attach themselves to a certain structure, you can really demonstrate that this cartoon, what you see there on the left, corresponds to what really happens in the cell in real time. I also like to show this example in particular because this is work that I did together in collaboration with my brother Niels Johnsson, who has been also very important for me in the development as a scientist. Okay, I could go on for hours giving you examples of protein labeling, and my wife can testify to that. But I don't want to abuse your patient. I also believe that showing you all these nice images gives somehow a misleading impression of like what we actually do on a daily basis in the laboratory. And most of the time what we do is we use synthetic chemistry to make molecules. And so I provided you also a picture of one of our hoods where the probes that I have just used are actually made. These hoods doesn't look as nice as the ones I've seen today in my tour here in the bio facility. But what I want to tell you with this is it is often synthetic chemistry that drives the innovation. Chemistry as the mother of invention, not only in chemical biology, also in different other fields. And so this as a reminder of how important chemistry is. The examples that I have shown to you so far focus on cell biology. But if you think about it, the possibility of attaching chemicals to proteins allows you also applications outside cell biology, for example, molecular diagnostics. The project or the example that I would like to give you concerns a disease that is called phenylketonuria, which is an inborn error in metabolism that leads to high phenylalanine concentrations in the blood. And if during development the phenylalanine concentration in the blood of a child are too high, that leads to intellectual disability. And so then what do you need to do as a parent or these patients is they need to have their phenylalanine concentrations regularly checked by going to the hospital, having blood drawn, and then analyze it using mass spectrometry, and then adapt their diet if needed. And if you think about it, it would be wonderful if for these kind of patients there would be a simple system available that would allow them to measure phenylalanine at home, such as you do with a fantastic glucose meter. However, such a device does not exist. And so we were interested in using the tools that we have developed to come up with a biosensor for measuring phenylalanine, potentially at the point of care. And so the outline of, or the design of the biosensor is summarized here on this slide. I do not really want to go into the details of it, but what it is in short, it is a luciferase, an engineered luciferase. Luciferases, you know, are the enzymes that are responsible for the emission from light from fireflies, for example, or deep-sea shrimp when they glow in the dark. And so we took this luciferase, designed a molecule that then we specifically attached to it through one of our texts. The molecule is shown there on the bottom, such that when now your biosensor binds to the analyte, the light emitted by the luciferase is changing from blue to red. And how this is happening, you can see on the right side of the screen, you see two Eppendorf tubes in which there is the biosensor in the presence and absence of analyte. And in the presence of analyte, what you see, phenylalanine is the analyte here, is you see emission of red light, whereas in the absence is blue. And so you can quantify the concentration of the analyte by determining the ratio of blue to red light. And this you can do on a simple piece of paper by spotting this biosensor on a piece of paper, then excluding external light by using this sophisticated box that has on its top a camera that then you use to take a picture. And the color of the emitted light, the color of the spot, together with the calibration curve, tells you what the concentration of the analyte is. Such a procedure has the potential to become suitable for point-of-care applications. And I show you now how in this paper that Patrick also referred to, we determined phenylalanine concentrations in patients. That is, you do a finger prick, you take a very small amount of blood, dilute it into a measurement buffer, and then pipette it on a piece of paper, from which you then take with a camera the color of the emitted light that then you use to calculate what the phenylalanine concentration is. And gratifying in collaboration with our colleagues at the Heidelberg, the university hospital, what we could see is that this simple approach in its precision is as good as the mass spectrometry that is currently used as the gold standard for measuring phenylalanine concentrations in blood or serum samples. I have to confess, of course, that this approach here at present is not yet suitable for patient self-testing because you cannot ask a patient to start pipetting him or herself. But what we're currently working on in collaboration with colleagues from the Fraunhofer Society is to try to find ways to integrate the different steps into a single chip that then hopefully we will be able to provide to patients at some point. I also show you this example because it is an example of experiments that have their origin in basic questions in chemistry or chemical biology, but then lead to something that has the potential of having a societal impact. That is the life of the PKU patients, that then they would be able to measure phenylalanine at home. With this, I would like to come to the end of my presentation. What I was hoping to show you is that the combination of chemistry, protein engineering, as well as cell biology allows you to create powerful tools to study biology. What I was also hoping to give you the impression is how much fun that is, this kind of interdisciplinary work at the interface of chemistry and biology. The final thing then is, again, thank you again to the Bayer Foundation for having me elected for this prestigious award. thank you again for my co-workers, for their fantastic work and working with me. That is so gratifying. I'm also very grateful to our society that invests so much in education and research. And this is just, I'm very grateful of this. And then finally, I'm grateful for your patience and looking forward to interacting with you now in the course of this day. Thank you.